Hello, everyone. My name is Austin Erie, and I am a biology PhD student at Texas A&M University in the Fitzpatrick Lab. And today I will be sharing with you some of my dissertation work looking at the sources of variance in female copulation rate in the Amboseli baboons. So across many species that practice internal fertilization, we see this trend in which females typically only copulate when their eggs are ready to be fertilized. And this is thought to be in order to maximize fertilization success while also minimizing the costs associated with copulating. So, reduce time to forage, harassment from mates, disease transfer, things of that nature. But we do see some exceptions to this trend. So, notably here, in that of the anthropoid primates. So these primates, like humans, have monthly ovarian cycling. And anecdotally, we do see that despite frequent mating, it can take females several cycles before they conceive, before they become pregnant. As an example here in the Amboseli baboons from previous work in my lab, here showing that pretty explicitly, it can take females up to 11 cycles despite frequent mating, supposed frequent mating in each cycle before ever conceiving. And so that leaves us with our big picture question being, why do females copulate and still fail to conceive? So one potential explanation for that comes from the sperm limitation hypothesis, which posits that when males are copulating with multiple females, the sperm supply of those males becomes constrained and ends up being a limiting factor for female fertility. And through that, we may predict that this would create a competitive environment for females in which they are competing for this sperm, and therefore females are going to copulate most frequently, and females that do copulate frequently are more likely to conceive. But before we can directly test or assess the relationship between copulation rate and conception, we first need to understand more about the copulatory behavior in general. How variable is it within a population, and what conditions are females mating more frequently in? So to do that, I am examining this in a population of savanna baboons known as the Amboseli baboons located in southern Kenya. They inhabit a semi-arid savanna habitat and they practice a multi-male, multi-female breeding system. So males mate with multiple females and females also mate with multiple males, even within one cycle. And also pretty important is that in these types of primates, females signal their ovulation, they signal their fertility using sexual swellings. And that is also a pretty big tool for us as researchers because it allows us to track when females are most likely to be ovulating. In order to assess these sources of variance, I am using data that was collected in 1994, 2000, and 2009, and 2010. So copulatory events were recorded for 66 Paris females. In conjunction, as part of the Amboseli Baboon Research Project protocol, daily demographic and reproductive information is recorded for all of these females. And so this provides us with plenty of sources of context to fully understand their copulatory behavior. So from the copulatory behavior, I calculated a copulation rate defined as the number of copulations per observation hour. So you'll see here sort of the range in my data set. So females copulate from 2 to about 15 times an hour on a given day. So you'll see that even though most females are mating from about 2 to 3 times an hour, it does get higher up, and you do see a good bit of variance. So across these 66 females and across these years, we ultimately have 306 unique female date combinations, or 306 copulation rates. So in this order to assess the variance in these rates, I ran a generalized linear model with copulation rate as my response variable, and I selected these 5 parameters seen here. So first being the female's age, second is CSR, or cycle since resumption. So this is an ordinal number indicating what cycle a female is on since resuming after her last pregnancy. So what cycle a female is on when that copulatory behavior occurred. Third being cycle phase. So as I previously said, we are able to pretty accurately track a female's cycle. And this includes a 5-day window in which ovulation is most likely to occur. We referred that as the late follicular phase, and the days before that 5-day window as the early follicular phase. And this has also been corroborated with hormonal support and previous work with this population of baboons. Our fourth parameter being Q. So this is the difference between the number of mature males and the number of cycling females in a group on a given day. So you could sort of think of this as a female's competitive reproductive environment, where a higher Q theoretically indicates a higher, more potential male mates a female has to copulate with. And lastly, we included a female's dominance rank in our model. Notably, we did not include female ID as a random effect, reason being due to our limited sample size and sort of some disparities within our data, ID was not represented across all of our parameters, and ultimately, its inclusion did not improve our model fit. To run our model, we adopted a information theoretic approach. A priori, all of these parameters were equally plausible in predicting copulation rate. And so this provides us a way to test essentially multiple hypotheses, to test multiple parameters at one time, where models with a lower AICC score indicates a more fit model. So here in this table, you will see our top 4 models. And in these models, we see that age, CSR, Q, and rank have appeared. In this bottom model, we have averaged the coefficients from these up here in order to get estimates per parameter. So you'll see that CSR and rank both were significant. And here under the estimates, you can see the effect sizes. So you'll see that age and CSR ended up having a negative relationship with copulation rate, whereas Q and rank was a positive relationship. Despite that, you'll see that each of these estimates were close to 1, indicating that these effect sizes are pretty modest. So ultimately, these aren't very strong predictors of copulation rate. However, what you will notably see is that in none of our top models did cycle phase appear as a predictor. In other words, females were mating equally frequently during the early follicular phase as they were during the late follicular phase. In other words, they were frequently mating or copulating even when ovulation was unlikely to be occurring. So again, females are most likely to mate or to be ovulating during this 5-day window. And overall, we sort of see that, or we do see that females, age, cycle number, Q, and dominance rank do appear in our top models, but these predictors are modest at best, sort of indicating that perhaps there are other parameters apart from the five here that could be important sources of variance and copulation rate. But again, notably, female baboons copulate as often in the early follicular phase as in late follicular phase. So perhaps it's the case that these copulatory events aren't strictly reproductive reproduction focused. It's not solely for the purpose of conceiving and becoming pregnant. Perhaps there are also um, um, social parameters, social reasoning, social context for why females are mating, perhaps during this early follicular phase. All in all, this does provide us context in going to our next step in assessing or testing the sperm limitation hypothesis. So from this, we'll actually be able to better assess the direct relationship between copulation rate and the probability of conception. So is it the case that females are more likely to conceive when they are copulating more frequently? And I look forward to being able to share those results at a future time. Thank you so much for listening.